How is it that we find ourselves surrounded by such complexity, such elegance?
The genes of you and me, the genes of you and me, are all made of DNA. We're all made of the same chemical DNA. We're all made of DNA.
Hi, you're watching DNA Today, a multi-award-winning genetics podcast where we explore everything to do with genetics, from CRISPR to rare diseases to new research. We have won the Science and Medicine Podcast Award for many years now. We have hundreds of episodes, and we really hope you enjoy these conversations where we dive into so many genetic concepts. I'm Kira Dineen, a certified genetic counselor, and your host. Whole genome sequencing, once a futuristic concept, has now become an integral part of the genetic landscape. When this groundbreaking technology first emerged, it came with a staggering price tag. Picture this. It's 2003 and the Human Genome Project has just been completed, costing nearly $3 billion to sequence a single human genome. Fast forward 20 years to today, this looks radically different. Thanks to remarkable advances in technology, the price of whole genome sequencing has plummeted to a fraction of what it once was. Nebula Genomics offers consumers whole genome sequencing and lifetime memberships for only $300. Yeah, that's it, $300. So I couldn't resist. I sent off my cheek swab to Nebula to have my whole genome sequenced for the first time. To explore these results with me, I'm joined by Kamal Obad, who is the co-founder of Nebula Genomics. Previously, he was a product manager at Google, working within Google Research and Chrome OS. Kamal was a Gates Cambridge Scholar at the University of Cambridge and is a graduate of Harvard University. Additionally, he's a Forbes 30 Under 30 honoree. Kamal, thank you so much for coming on the show. I am so excited to dive into my genome, which is really cool because I've never done this on an episode of DNA Today. So I'm very excited. I even kept my kit here to show off to people that are watching the video of kind of what that looks like from Nebula Genomics. So thank you so much for coming on.
Yeah, thanks so much for having me. I'm super excited to go through your report with you.
So I first learned about Nebula Genomics from its association with Dr. George Church. And for those that don't know, he's the founder of the company. Is it co-founder or he's the founder? I want to make sure I get that right.
So George is one of three co-founders of the company. So it's myself, George, and one of his old PhD students, Dennis Grishin.
Very cool. So the three of you started this company, and for those that don't know, Dr. Church's pioneering work has contributed to the development of DNA sequencing and genome engineering technologies. I mean, his CV is quite long, 550 publications, 150 patents, wrote a book. He started over 20 companies. So I'm really intrigued. We're talking about Nebula Genomics today. Why Nebula Genomics? How did you get involved with Dr. Church as well as your other co-founder? What was the inspiration to say, we need to start this company specifically as opposed to some of the other companies that he's been involved with starting?
Yeah, so we started the company in around 2018. And around this time, had been a lot of adoption around consumer genomics. And at the time, I wasn't working in the genetic testing space at all. I was actually working at Google at the time. But I had always been familiar with George's work. I'd been to talks and lectures that he's given and read his book. So I've always been a fan of what he was doing, and I've been particularly interested in the genetic testing space. So back in 2018, consumer genomics had really taken off, had become a household name. But kind of one of the observations that we made was that despite consumer genomics having become something that everybody knew about, or a lot of households have done, it didn't really have a huge or meaningful impact on your actual health and wellness. So it wasn't being used clinically as widely. And at the time, I was pretty naive about this and wanted to understand why that was. And one of the big reasons I settled on was, maybe the type of genetic testing that are being turned into products that are being commercialized to mass markets aren't as powerful as they could be. So a lot of these companies were using things like genotyping or SNP chips essentially to actually do the genetic testing. We thought that you could do whole genome sequencing. So around this time, I reached out to George. I reconnected with him. I met with him and started discussing what a next generation personal genomics company would really look like. And some of the things we really hit on were a next generation personal genomics company will generate the highest quality data. So for us, that meant clinical grade whole genome sequencing. And at the time, we were kind of like right on the precipice where whole genome sequencing was too expensive for consumers, but the price had been going down really quickly. So we were optimistic about the direction the price was going to go. So that was one big thing for us, was really high quality genetic data. The other one was a big focus on privacy. So less focus on monetizing data or licensing data to pharma companies, and more emphasis on building a product that consumers and patients would actually benefit from, that would align with them. So that was the original idea around Nebula, was take the opportunity, the tailwinds, so to speak, from kind of the adoption of consumer genomics and the interest, but shifted away from some of the ancestry applications, which are more like entertainment, very consumer-friendly infotainment type products, and build a next-generation personal genomics company that was really more focused on the health and wellness applications, built around the idea of commercializing whole genome sequencing. So that's kind of how the whole thing started. I met with George. We had very similar ideas and it's very similar sense around where the space was going. And it turns out he had a PhD student in his lab, Dennis, as well, who felt very similarly. So we all decided to team up and launch the company.
Wow. And it must have been such an interesting time in 2018, because other direct consumer genetic testing companies in the space were very much household names, as you said, but they were offering testing that was a little bit more entertainment, as you said, some health information for some of the companies, but also limited. I don't remember any other company at the time being like, we're going to offer whole genome sequencing where you can order it yourself and you don't need to be going to a doctor's office to do that. So
yeah,
very, very different, and just giving so much more information. And a lot of people listening are probably familiar with what a whole genome sequence is, but I think it's also helpful to compare it to other genetic testing and the benefits of having access to one's entire genome compared to other more limited genetic tests like gene panels or even exomes, of just how much more information you can get and how that can be beneficial, because it really is a lot more information even compared to an exome.
Yeah, so I think at the time, so when we were starting Nebula, historically doing whole genome sequencing was prohibitively expensive for consumers. So it's kind of like what you mentioned. It was really a premium product, really edge cases. People would do it with doctors. They used to be spending thousands of dollars out of pocket to do it. But we thought the benefits of whole genome sequencing were really exciting and also necessary for genetic testing technology to actually have the impact to patients that we hope it's eventually going to have one day. And I guess to give like a high level summary of the different types of technologies, your genome is really, really long, right? It's billions of base pairs, essentially billions of letters long. What we strive to do with whole genome sequencing is essentially, at a high level, read your genome almost end to end. So almost like we're reading a book cover to cover. We're really trying to get your entire whole genome sequence. And what most people realize is that the vast majority of genetic tests that they've heard of don't actually do this. The way they actually work is they pick and choose specific regions or specific parts of your genome that we know, if you have specific variants or differences there, could have an impact on your health. And that's how most of these tests work. And one of the biggest constraints and reasons for that was really around cost, was just the cost of doing a whole genome. So a lot of the popular tests do like a DNA microarray or a SNP chip. You pick and choose maybe 500,000 variants that are well documented. They're in the literature. We know what they do. Exome sequencing, it's a step up. You're sequencing all the genes. So you have about 20,000 genes in your genome. Instead of sequencing your entire genome, we can sequence just the genes, but that's still like a low single-digit percentage of your actual DNA. What we do with whole genome sequence is we really try to get 100%. The challenge of that is we don't know what 99% of your DNA does. A lot of it is as far as we know, is in non-coding regions. We don't know the functions of different variants. And the reason we don't know is that historically there hasn't been a ton of whole genome sequencing data that's existed for us to actually study and analyze. And that's really quickly changing as the cost goes down. But the important thing about whole genome sequencing, to kind of get to the core of your question, is we've realized over the past 20 or so years that the way genetics affects your health is largely polygenic in many, many cases, especially to the average individual. Yeah, so you're not going to find one gene that tells you, or one variant that tells you some life-changing thing in most instances. Those instances where that does happen are very rare. But instead, what you can find is many, many genes that each have small incremental effects on particular traits. So we find that a lot of traits related to your health and wellness are polygenic. And we're still fully trying to understand how these polygenic scores should be calculated and how they should be reported. But you really need whole genome sequencing data or really wide breadth data to actually generate these kind of polygenic scores that can be much more meaningful to the average person.
Yeah, and it's so much data to go through. From the research I was doing, it seems like whole genome sequencing provides about 100 gigs of data, but that probably also depends on the quality of the data. I don't know if I'm even close in a ballpark there. How does Nebula Genomics help users actually make sense of all of this data and information? And as you brought up, relevance to their health, because there's a lot of aspects, and we're going to get into mine after this part, of just, okay, this is what we're analyzing of your risk to develop this condition or this health concern. How do you take all of these gigs of data and actually go and say, this is health risk? That's a lot to process.
Yeah, so I would say, it's still a work in progress for us. I think that's the big challenge of the field, is taking these, and you're right, like around 100 gigs, sometimes more. We do also 100x whole genomes, so sometimes 300 plus gigs, is taking this large amount of data and generating interpretable insights from it. So that's still a work in progress. A lot of our patients or customers, they actually take that data and they go work with a clinical geneticist or a genetic counselor to get expert insight, and we provide easy to use tools to actually navigate and browse your genome. So what we do is we provide like a high-level report. We call it the Nebula Library. I'm sure we'll show what that looks like. That gives summary statistics around different traits and really links these types of traits to what the literature is saying. We also give much more powerful tools that we've seen a lot of our customers use, where they work with a physician or an expert or a genetic counselor, for instance, and they actually go into this gene analysis tool where they're able to browse specific genes or generate synthetic gene panels and look at the types of annotations that different variants might have. In terms of automating this end to end, we're not quite there yet. The way our reports work today are not diagnostic. We essentially generate all the data and we provide you with all the data if you have the expertise and skills to go and explore that, or you want to work with someone to explore that. But we also provide easy to use web-based tools to actually browse this. And we've tried to make these have like a range of required sophistication, I guess, to use these tools. There's, we even have like an IGV browser that's built into the website. We have easier to use tools, gene analysis tools, like I mentioned, the gene panels, and then we have high level reports.
Yeah, it's amazing just how much information we can get from this. And a lot of it is what we mentioned of like polygenic risk scores, of looking at multiple genes, multiple variants in those genes and saying, well, based on the accumulation of this is what we're estimating your risk to be to develop a condition. And so, I figured we could dive into my data, because going through, I was just like, as I go through my data, I'm just going to write all these questions and then Kamal hopefully can answer them. So we'll see. So I sent in, as I showed for people watching the video, this is the kit that I had. And so kind of held on to it because I figured, video, we'd show it on the camera here. So I did a cheek swab, got my results in less than a month, which I was very surprised by. I wasn't expecting results that soon. So it was a Saturday morning. I woke up, got an email saying my results were in. So it was a nice day outside, sat on my patio with my coffee, started exploring. And I have to say, it was quite fitting as I'm drinking my coffee to read, I have above average genetic predisposition to higher coffee consumption at 87%. And as I'm sipping, I'm like, yeah, that is very fitting. That is scientifically accurate, I would say.
I'm glad that was accurate.
Yeah, that was, I feel like I should have been like, on The Office, like where you look at the camera and you wink or something. You do one of those gym looks, right? But how do you guys come up with these risk scores? How much data goes into coming up with this score? Like, it's very specific, right? I have a higher predisposition to coffee consumption. I'm 87th percentile, so I'm really up there. Mm-hmm. How do you figure that out? Or how many different variants are you looking at for traits like this that, this one isn't really health related. It's just more interesting, right? what is the process to say we're going to find certain variants, and what do you include, what do you exclude, what's required to be on that list?
Yeah, so those reports in the Nebula library, we have a team of scientific writers led by our chief scientific officer, who's a genetics expert, Dennis, who's in George's lab. And the way those reports work, we curate the reports. We don't curate the actual variants that are included in the report. So those reports, every single entry in the report is based off of a peer-reviewed paper. So high quality research that's been vetted, that's been peer reviewed, where they've done some sort of GWAS analysis or statistical study on how different types of variants can affect specific traits. And that gives us effectively, from these papers, we get a list of variants. So these are differences in your genome from the reference genome. And we effectively get effect sizes and how they could impact that trait. So using that information, we can generate a summary statistic, again, just related to that paper. So if there's a paper on coffee consumption, they found a bunch of genetic variants. We'll tell you what that paper means within the context of your genomics. So we generate a summary statistic, and then where the most interesting part of the report comes in is how do we compare that to the rest of the population? So how do you know if your score is high or low? And that's built on our existing data set. So we have upwards of 100,000 people that have had been sequenced or genetically tested with us. Those folks are incorporated in the data set, and they form our reference data set. So we compare you to that existing population of users to show you your percentile. There's a lot of caveats still with the data. That's why we always stress that it's not diagnostic. It's not necessarily prescriptive. Because one is, what do these scores mean? So one of the comments we get a lot of time is, I have a 99 percentile score. Does that mean I have a 99% chance of getting this? That's typically not, that isn't what it means. So this is just within the context of your genetics and this paper, we can tell you what percentile are you at in terms of the genetic contribution to this specific trait. So for some traits, for instance, to kind of simplify that, like your love for coffee, that might, 90% of your love for coffee might come from just, you've had a lot of good coffee in your life, and you've shared some good conversations over coffee, so you really enjoy coffee. 10% of that might be genetics, right? So 90% of it's environmental stuff, 10% of it is genetics. So there's really only a small part of why you love coffee is explained by your genetics. That's the same for things like obesity. It might not be a lot of genetics. There might be a lot of environmental things that play a role as well. So we're just showing you the genetic component for those traits in the library.
Yeah, it's really interesting. And I'm now going off of memory, which is a scary thing. But I seem to recall at the end of those pages when I clicked on the coffee to read more, you have either links to the papers or those references. So for nerds like me that want to look further into it and say well, how did they come up with this? How did we figure out those variants? And you mentioned GWAS studies. For those that don't know, those are genome-wide association studies. So looking to see how does this variant, this spelling change in someone's DNA play a role in whatever trait you're looking at. And it's really interesting. I think that was the one that I just kind of, laughed at as I opened it, but laughed at in a way of so accurate. And there are loads of categories like autoimmunity, behavior, cancer, cardio, metabolics, pregnancy. How did you guys figure out what traits you actually wanted to include? Do you have a requirement for how much data you need to be able to back up those percentiles that you mentioned?
The way we make the decisions is, one, we start with the quality of the actual research. So is this high quality research? Was the GWAS or analysis performed with large sample sizes? How was the study conducted? Is it peer reviewed, for instance? Are there other studies that replicate or show that the variants that were detected are meaningful or are accurate? So we have a checklist of different requirements before we include reports in the Nebula library. And our filters for what a high quality report is typically means that we're adding a handful of reports per month to the library. So sometimes 2, sometimes 3, sometimes 4. So we're pretty strict on what actually gets included. We also have reports that are, different papers but around the same trait. So there might be two entries in your Nebula library for a similar trait, but the analysis comes from different papers with different variants. So you can compare how different papers are looking at the genetics differently and what your scores are for different ones. But yeah, that's a high level summary. It's really led by a team of our PhD scientific writers and just expert curation of what we think is a high quality resource.
And you hit on an interesting point that this is not static information, where on this Saturday morning when I was looking through my report, I was like, okay, like that's the end of that. This is something that I'm continuously throughout my life going to like look back on and see like what else you guys have added. And that you mentioned at the beginning, this is data that you guys have, sequenced for me. And now I can take that and use it however I want in the future, which is really interesting. As someone that's a genetic expert, I can kind of go to people that know even more about genetics, know more about sequencing than me, and look at this data together, which I think is really exciting. And for those that, are genetic nerds, and you probably are if you're listening to this, it gives you that flexibility just over the course of your life that you can kind of keep coming back to this.
I think that's like the big benefit of whole genome sequencing is kind of our hope is, sure, we only know what a small percentage of your DNA does today, but we're going to learn more over time. There's tons of progress in understanding how, non-coding regions can affect your health and wellness. And those reports will be added to the report over time. So we keep giving updates regularly to everybody.
Yeah, so I'm in my late 20s, so I hopefully have many decades to go where it's just like going to explode in terms of all the information that I'm personally going to benefit from looking at all this, as well as my relatives too. And, you start spanning out in terms of, these genetics didn't just magically appear in me. So, first degree relatives, second degree relatives, and all of that. One of the interesting aspects too, in terms of types of traits we look at, there's a behavioral trait section, and gambling is one of them, which also is one that really accurately predicted for me that I have a very low percentile for this. I was 2%. And that's something that matches up with me. I'm very conservative when it comes to finances. I don't, I spend a lot of time in Vegas, but I don't know if I've ever gambled. So I think that kind of backs up my story a little bit there. How do you, or these research articles and peer-reviewed journal articles that you're linking to, how do they come up with this? Because that's hard to look at. If we look at things that are what we can measure of someone in a very non-biased way, that's a little bit easier. But are you aware of these studies being exclusively like self-reporting, of people having a survey of they submit a sample and then they're looking at the genetics and saying, oh, are you someone that gambles more or less? I'm just wondering kind of how studies like that work, if you're familiar with them.
Yeah, that's a good question. So that one specifically, I actually don't know the details of how that study was performed, but I think it's a combination of things. So sometimes it's retroactive studies on collected clinical data or self-reported survey data. And if you have big enough sample sizes, you can glean significance as long as the study was done well, even if it is self-reported. The thing that's interesting today is, which wasn't true not too long ago, was that there's a lot of access to really rich genetic data and clinical, phenotypic, or behavioral data, whatever, all these different types of complementary data types. There's a lot more that's widely available through different types of initiatives, public data sets. UK Biobank is a great example, and these have just really come online over the past few years. So that's why we're seeing an acceleration of how these types of studies can be done and the scale that they can be done at, because that's the hardest part about these GWAS studies, is large enough sample sizes to actually find variants that look like they could potentially be causal or have big enough effects where they look like they could be causal.
And hopefully that will be less of an issue as time goes on with projects like the Human Genome Project, of just deeply sequencing a lot of people in the world, where I believe the requirements for that is, we'll put a link in the show notes in case I'm wrong in this moment, but where someone's 4 grandparents are from 4 different places in the world, ancestry-wise. So I think we'll hopefully have a lot of variants that we have curated at that point, and we understand it much more just as time goes on. I think we're going to see that just in our careers. When it comes to diet, this was an interesting aspect of my report. I have a higher predisposition to, consume more protein, fat, carbs. So I'm kind of like, okay, that's a lot of food. But lower predisposition when it comes to sugar. one aspect of this that I'm a little bit more worried about when I look at it, of like, maybe this should motivate me to do something about my health, is I have a very high genetic predisposition, which, as I'm saying this, I'm a high percentile. I should change my verbiage with this, to higher HDL cholesterol levels and to develop type 2 diabetes, which is a little bit surprising because people in my family, as far as I know, and I have a fairly big family on both sides, nobody has been diagnosed with type 2 diabetes. But wondering, when you talk with people that have done genome sequencing through Nebula Genomics, of what do conversations look like in terms of next steps? Obviously, you're not my doctor, you're not going to be able to tell me do this. But what steps do people often take, or to look at lowering that risk? Because knowing that if genetics is playing this role, what can I do in terms of that other aspect? And just, I guess, with type 2 diabetes, like, how much do we know about type 2 diabetes when it comes to the influence of genetics versus environment? Because obviously it's going to be a playing of the two. Yeah,
it's interesting. I mean, type 2 diabetes is an interesting case, specifically because it is a huge part of it is genetic, even more so than type 1 diabetes, which a lot of people think, it's the opposite.
I thought the opposite until not that long ago when I learned that. I was very surprised.
Yeah, it's surprising to most people because type 2 is something that you think of, developing, right, over time, but there's a huge genetic component to actually developing it as well. But in terms of making things actionable, I mean, most of the customers and patients who use us, I would say a big chunk of them do have a question they're looking to answer. So we've had a lot of patients they're in the rare disease space, maybe they have an undiagnosed condition. They get our data and they work with a third party to figure out a diagnosis. We have a lot of people who have a family history of something. They're interested specifically in diving into that more deeply and looking at their genetics of it. So I would say a lot of folks use it as a conversation starter to have conversations with their doctors, their provider, perhaps whatever expert resource they might have at hand to figure out what the implications can be of different types of traits. The thing we stress pretty well in the reports is, these are like not diagnostic. These are not telling you you're going to get something, but it can inform you to start a conversation or think about something a little bit more and figure out what you want to prioritize or what you want to optimize. But really, it gets complicated. I think our reports could definitely even do a better job of sort of trying to explain how even if you have 100th percentile chance of something, that's just your genetic predisposition based off this one study is at the 100th percentile. That doesn't mean you have a certainty of developing. You might still have a very, very small chance of manifesting whatever trait the report's about. So most of the reports are best used as the beginning of like a thread to pull on. They're best used as a conversation starter or as like a first step before a deeper analysis is actually done.
Yeah, I think that can be helpful of something that's not top of mind, being, a youngish, healthy person. But now I'm like, should I be, looking a little bit more into that and just having it be mindful about that. And like you said, I was looking at some of the cardiac information, the cardiovascular health section, and a lot of it I had a high chance, or, I'm in that high percentile for genetic chance of developing conditions like hypertrophic cardiomyopathy, coronary artery disease, and I was the 100th percentile for longer PR interval. So like that's something that it's not meaning that I will develop that condition, but it's just like, do I have more of a chance than other people when it comes to that genetic component? I would think yes. But along those same lines, like I'm predicted to have a higher blood iron level. At the same time, when I go to donate blood, I am often turned away because I have low iron, right? So it's also, there can be times where we're looking at some of these polygenic risk scores and it doesn't line up because there isn't a ton of data behind that one trait. And I was diving a little bit deeper into this aspect because I'm like, this doesn't line up. But then it made more sense because it looks at, I don't know if it was 6 genes or variants, probably variants, were calculated for this. So now I'm going to kind of look at it and be like, well, let me check back with that, because over the years, I'm expecting more and more variants to be added to that. And then maybe that genetic percentile is going to shift over time as more variants are added. How often do you recommend for people to check back with this in terms of updates. I don't know if you guys are just on a rolling basis updating or if it's more like quarterly or semi-annually. What do you recommend for people, especially that it's not, it's just kind of like they're just checking in and they're not looking for a diagnosis or anything along those lines?
Yeah, I would say we send, at least monthly emails with kind of a summary of what the updates look like. So that's probably a good percentage of our users check in about once a month in the reports, typically when those monthly updates are going out.
Yeah, definitely. Is there any on the horizon that you're able to share, or is that just, private information and we have to subscribe to the newsletter to stay tuned?
Yeah, you might have to subscribe to the newsletter, but I actually just don't know on the top of my head, unfortunately.
That works. Stay mysterious. Then people will subscribe to the newsletter, right? So now that we've processed a lot of my results, hopefully people are intrigued just to see like where they fall on this. I have to say I had so many questions going into this, and I am so happy you were able to answer so many. If we had more time, I would get into even more because I got very overzealous about preparing for this interview. But now that I have my results and we've processed it a little bit more, what should I talk to my family members about regarding these results, or other people listening that maybe are going to be doing whole genome sequencing, or maybe they have before and they saw Nebula Genomics and they're like, oh, let me check in because I actually have already done a kit with you guys. What comes to mind when, looking at reports of maybe aspects people should pay attention to in terms of having conversations with biological family members?
Yeah, I think the best thing to do is look at things that might be concerning to you in the report. And again, if you get this report, there is nothing that we're telling you that is going to completely change your life. There's nothing terrifying that we could possibly tell you in the report, but there are informative things. So I would recommend taking the report, finding things that stood out to you, and having a conversation with your parents or your grandparents and seeing, is there a family history of this? Is there something we haven't talked about before? Is this a conversation I should have with my doctor? I think those are all fair starting places because that's one of the most important things that doctors and providers do is look for your actual family history and try to understand. That's kind of a way to try to analyze how your genetics could impact your health, but we're just giving you even more information on top of that. So I think that's a great way to really use the data that we provide.
Yeah, as a genetic counselor, I was kind of hoping you would say that, about plug to talk with biological family members if you have them available and have those relationships to explore family health history, because that is, I still think, the best genetic test at this point in time of looking at how has genetics played out in many people in your family that share these genetics. Mm-hmm. Because if there are conditions, traits that are being identified through you guys with Nebula Genomics, and then you talk about it and they're like, yeah, most people in our family have actually had this issue, that might be enough of a motivator to go see a healthcare provider to have those conversations for management, prevention, treatment, whatever is appropriate for you. So I think it's great to have this available to people, that they can order this directly, get this information. But I think that's also good advice that you've given throughout the interview here of, as you are learning about things and if you are worried about aspects of this, that like the next step is, well as talking to your family members, is a healthcare provider, that if there is something that you're like concerned about. But this was great. I think a lot of people are probably very intrigued now to see where they fall in the percentiles for all of these different traits and more. So if people do want to have their whole genome sequenced by Nebula Genomics, they can use code DNA Today, one word, DNA Today, name in the show, easy to remember. And you guys have been very generous to offer 15% off. And on top of that, you guys are doing a giveaway with us. So if you would like your own kit, head over to our social media at DNA Today Podcast on Instagram and LinkedIn. Directions are on there to enter. And thank you so much, Kamal, for just going through my report with me. This was really interesting. And like you said, a lot of this was, my curiosity, I think was, very much met in terms of what I'm learning from this. But there also wasn't any information that I was like, freaking out about or felt like I hadn't processed enough ahead of time for that like pre-test counseling that we talk about as genetic counselors a lot. I was able to finish my coffee. I wasn't starting to have anxiety where I had to abandon that, which, happens in life for people with anxiety issues. So, but thank you so much just for going through all this. I think it's really interesting, and we just covered part of it. So I think for people to check it out, and if they have more questions, you guys can email into the show, info@dnatoday.com. But thank you so much, Kamal.
Yeah, thanks so much for having me. This was a lot of fun.
Thanks for watching DNA Today. To access all of our episodes, head over to dnatoday.com. We also have a lot of bonus content on there that you can enjoy. If you have any questions, comments, suggestions, guest pitches, you name it, send them in to info@dnatoday.com. We'd also really appreciate if you could take a moment to rate and review the podcast on Apple, Spotify, wherever you listen to the show. It really helps more nerds like yourself find the show. Also, if you like giveaways and other ways to connect with us, I recommend following us on social. We're at DNA Today Podcast. We also have a Patreon if you want to be the most level involved in the show. That's also at DNA Today Podcast. Thank you so much for listening and watching. Join us next time to learn and discover new advances in the world of genetics.
We're all made of the same chemical DNA.